Hello and welcome to tutorial 25. Our topic for today is nucleic acids. Before we get started, just a quick reminder that the notes to accompany this video tutorial are available online through my website. And with that said, let's go ahead and get started. Nucleic acids carry all of an organism's genetic information. And there are really two subcategories for nucleic acids. There's deoxyribonucleic acid better known as DNA. So DNA. And there's ribonucleic acid, better known as RNA. RNA. DNA carries an organism's entire genetic code, and the DNA is stored within the nucleus of most cells. RNA contains just a copy of one gene from the DNA. And it's mostly found outside of the cell nucleus in the cell cytoplasm, where it is involved in protein synthesis. Now, for this tutorial, we are focusing specifically on the structure of nucleic acids, DNA and RNA. In our upcoming tutorials, we're going to focus on the function of DNA and RNA. Specifically, we'll be talking about DNA replication, transcription, and translation in those upcoming tutorials. So, focusing just on the structure today. Nucleic acids are polymers of smaller molecules known as nucleotides. And so, since we're interested in structure today, it makes sense that we would start with the structure of nucleotides, those molecules that link together to make up those nucleic acids. Okay, so I'm going to write up here, nucleotides. And nucleotides are really composed of three parts: sugar, a nitrogen-containing base, and a phosphate. So I'm going to write that up here. Composed of three parts: a sugar, a nitrogen-containing base, and phosphate. Okay, so we're going to start with the nitrogen-containing base component first. So nitrogen-containing bases. And you'll notice that I've actually already drawn all five of the nitrogen-containing bases found in both DNA and RNA up on the board. And I've also have those drawn out in slide 3 of the notes. Okay, so hopefully you have those in front of you already. Let's switch to a different colored pen here. So we have adenine, better known as just A. It's abbreviated as just a capital A. We have guanine, capital G. Cytosine, capital C. Thymine, capital T. And uracil, capital U. Now you'll notice that adenine and guanine have a very similar structure. That's because they are both derivatives of a molecule known as purine, and so they are often collectively referred to as the purines. Okay, so I'm going to write that here. Adenine and guanine are your purines. Notice a similarity between cytosine, thymine, and uracil. That is because they are all derivatives of a molecule known as pyrimidine. And so they are often collectively referred to as the pyrimidine bases. So I'm going to write here that these are our pyrimidines. Now, adenine, guanine, and cytosine are found in both DNA and RNA. I'm going to write that here. A, G, and C found in both DNA and in RNA. But thymine is only found in DNA, and uracil is only found in RNA. I'm going to write that down as well. T, only found in DNA, and U, uracil, is only found in RNA. So another way to say that is that adenine, guanine, cytosine, and thymine are all found in DNA. And then when the copy of RNA gets made, all of the thymine gets replaced with uracil. All right. Okay, so we talked a little bit about those nitrogen-containing bases. Let's next talk about the sugar component of the nucleotides. Okay, so let's go back to the black pen here. And I'll underline that just to make it stand out a little bit more. We'll do sugars. Okay, the sugars. In RNA, the sugar is always ribose. In DNA, the sugar component is always deoxyribose. So let's write that down here. In RNA, the sugar is ribose. And I'm going to go ahead and draw its structure here. And then in DNA, the sugar is always deoxyribose. Very similar structure, but as the name implies, deoxy, it's going to be missing one of the oxygens. In particular, it's going to be missing the oxygen at this 2 position. Remember that the anomeric carbon, this hemiacetal carbon, is always position 1. So this is position 2, 3, 4, and 5. So at position 2, we're going to be missing that oxygen. We're just going to have a bond to the hydrogen in deoxyribose. So other than that, the structures are the same. It's kind of a messy oxygen. So don't put an OH here. Skip that position. That's the deoxy position. And remember, this is a line structure, so we don't show our bond to hydrogen here. Okay, so there's deoxyribose. And probably not surprising to you at all that both of these sugars are going to be D-isomers. So I'm going to write that here. Because as you might recall, nature is selective for the use of D sugars and L amino acids. Okay, so not surprising that the sugars involved in DNA and RNA are going to be of the D isomers. All right, the board is quite full, so let's go ahead and take a moment to erase before we go on. When a nitrogen-containing base is linked to a sugar unit, the result is a nucleoside. I'm going to write that up here on the board. A sugar plus a base, I'll just say a base, equals a nucleoside. Not to be confused with a nucleotide, with a T here. Because remember, we just said that a nucleotide has 3 components: the sugar, the base, and phosphate. When you don't have that phosphate yet, it is a nucleoside. It's a smaller molecule called a nucleoside. And the sugar and the base, let's go ahead and underline that there. The sugar and the base will be linked together by a beta-N glycosidic bond. I'm going to write that up here. Linked by a beta-N glycosidic bond. And I'm going to show an example of that here. Let's do the sugar deoxyribose. So I'm not going to put an OH here. And then I'm going to show my N-glycosidic bond to the nitrogen-containing base. Let's pick an easy one to draw. Let's do cytosine. And you may want to clarify with your instructor whether or not you need to know the structures of the 5 different bases. When I was teaching introductory biochemistry, I did not ask my students to know those 5 specific structures. So again, clarify with your own instructor whether or not you need to draw these from memory. Okay. So I'm going to go ahead and just do one of the pyrimidines because they're a little bit easier to draw. I'll do cytosine. It's a nice, easy one to draw. I mean, relative to the others. All right, so let me just check that structure real quick based on what I have on slide 3. I wouldn't want to make a mistake there. Okay, so I've got the base cytosine linked by a beta-N glycosidic bond to the sugar deoxyribose. So this is a nucleoside. So let's highlight this bond here. This is our beta-N glycosidic bond. And you might notice already the similarity to the glycosidic bonds that we saw form between 2 sugars to make up a disaccharide. Remember that the hemiacetal reacted with an alcohol in that case. Well, in this case, the hemiacetal is reacting with that NH off of the cytosine, the pyrimidine base. And so that's why we call it an N-glycosidic bond, because it goes to a nitrogen instead of an oxygen there. And the beta position, recall, refers to the orientation of that bond in the upward direction. In other words, on the same side as the CH2OH of carbon 5, okay? So if it were pointing down, because we know that this is a tetrahedral geometry, it could go up or down. In this case, a hydrogen is down, but if we switch those, then that would be a glycosidic bond, an N-glycosidic bond in the alpha position if it were pointing down and were opposite of the CH2OH. So that's why it is a beta with respect to the orientation in the upward direction, the same side as the CH2OH, N-glycosidic bond, because a glycosidic bond goes to a nitrogen. All right. Let's go to the pink pin here because I really want this to stand out. Notice that we have multiple ring structures here. So when we count, we're going to count the carbons, or the atoms I should say, in the ring structure of the nitrogen-containing base, and we're also going to count the carbons in the ring structure of the sugar. Okay, so I'm going to number these 1, 2. I'm going to count through these higher priority groups, the nitrogens and the carbonyl. 3, 4, 5, and 6 would be the numbering for our nitrogen-containing base. And then when we go to number our cyclic sugar, we're going to use a prime to indicate that it is a different ring than the nitrogen-containing base. So we've got a 1' for the anomeric carbon, 2', 3', 4', and 5'. Okay, so that's how we would number our sugar, all the carbons in our sugar. Okay. All right, so let's talk a little bit about how to name nucleosides. Because if you can name a nucleoside, you can definitely name a nucleotide. In fact, naming a nucleotide will be very, very easy for you if you already know how to name a nucleoside. So let's go ahead and do the hard part first. And then again, it'll be very easy once we get to naming nucleotides. So it's right here, naming nucleosides. Okay, and when we're naming nucleosides, our name really needs to tell us two things. It needs to tell us what is the sugar and what is the base. So, what sugar and what base? Because those are the two components of a nucleoside. Okay, so let's start out with the sugar component. If the sugar is deoxyribose, then you're going to use the prefix deoxy. Use prefix deoxy. If the sugar is ribose, do not use a prefix at all. No prefix. So the lack of a prefix in a nucleoside name tells you that the sugar is ribose. Okay. The base. If the base is a purine, then we're going to use the ending OSINE. I'm going to write that down here. For the purines, use the ending OSINE. So what does that mean? That means that the base adenine, once it's linked to a sugar and it's become part of a nucleoside, we're going to change the ending again to OSINE, and the name becomes adenosine. The other purine, remember, is guanine. So as a nucleoside, you'll use the ending OSINE, and it will become kind of a messy G. It will become guanosine. Okay. For the pyrimidines, you're going to use the ending IDINE. Okay, so specifically, that means that the base cytosine, which is a pyrimidine base, becomes cytidine as a nucleoside. The base thymine, the name changes to thymidine for the nucleoside. And last but not least, uracil becomes uridine. as a nucleoside. All right, so let's go ahead and name the nucleoside that we just happened to draw up here on the board a moment ago. Let's go to the blue pen for that. Well, let's make it stand out. Let's do green. Okay, so we know that our sugar in this particular nucleoside is deoxyribose. You can see it's missing the oxygen at the 2 positions. So it is deoxyribose. The base, again, your instructor may or may not want you to memorize a specific structure for all 5. If you don't know the specific structure, look it up. I have them drawn on slide 3. You'll notice that I happen to have drawn cytosine. Okay, so the base is cytosine. Okay, so let's put those together. We get the prefix deoxy, and cytosine becomes cytidine, deoxycytidine. That is the name for this nucleotide that we just happened to draw above in our example right here. All right, so real quick before we go on, what if this were ribose? What if there were an OH here? Well, then we wouldn't have the deoxy prefix, and we would just call that nucleoside cytidine. You would know it was a nucleoside because of the ending for the base. This pyrimidine base has to have the ending idine in a nucleoside. If it were just the base and it were a nucleoside, you wouldn't have changed the ending. So the fact that the ending was changed to idine, you know that that means that it's a nucleoside. All right, let's erase here and go on to nucleotides. Okay, so on to nucleotides. A nucleoside plus a phosphate gives you a nucleotide. I'm going to write that up here. A nucleoside plus a phosphate gives you a nucleotide. And that phosphate is linked to the nucleoside at the 5' position via a phosphate ester bond. I'm going to write that up here. Linked to the 5' position by a phosphate ester bond. And I'm going to show that. Okay, so let's just stick with the nucleoside that we saw previously. So that was deoxycytidine. Okay, so I'll draw my sugar, deoxyribose, but I'll leave room to add my base and my phosphate. So let's go ahead and add our base first. Okay, and I'm going to double check it. Just want to make sure that I don't make a mistake there in my structure. Okay, and I'm going to just check it with slide 3. So that is the structure for cytosine, the base cytosine. Taken together with the sugar, it makes a nucleoside deoxy, for the deoxyribose, deoxycytidine. We're going to go ahead and make it a nucleotide by then linking our phosphate here. Okay, and I'm going to label that phosphate ester bond. So we've got our phosphate ester bond here. Okay. So if you might notice that it looks like an ester. An ester, remember, has a carbonyl connected to an oxygen and then connected to some R group, some other number of carbons. So you'll see that similarity here. So our phosphate ester linkage, I'll highlight it right there. Okay. And we said it happens at the 5' position. Remember, that's because when we number the sugar, we use the prime to distinguish them from the numbering system in the base. So we had a 1', 2', 3', 4', and that bond happens off of the 5' position with the alcohol off of the 5' position. Okay, so now we have a nucleotide. So how do we name nucleotides? Well, the good news is if you understand how to name a nucleoside, this part will be very, very easy, because all you do to name a nucleotide is first name the nucleoside and then add 5' monophosphate. That's if there's only one phosphate. If there's 2 phosphates, it's a diphosphate. And if there's 3 phosphates, you'll add triphosphate. So let me write that down here. Naming nucleotides. Name the nucleoside component and then add 5' monophosphate. If there's only mono or 1 phosphate, if there's 2, then it's diphosphate. And if there's 3, then it is a triphosphate. Now, in truth, a lot of textbooks will leave out the 5'. Okay, so I'm going to make a little note here. Often left off. Okay, but in case your textbook shows it, I just wanted you to understand where the 5' is coming from. It's telling you it's off of the 5' position of the sugar ring in the nucleoside. All right, so let's go ahead and name the nucleotide that we've drawn up here. And let's do that in green. Make it stand out a little bit. Okay, so in this nucleotide, we have the sugar deoxyribose. Okay, notice we're missing the oxygen at the 2 position. There's just a hydrogen there. We have the base cytosine, and our phosphates, we only have 1, just 1, so it's going to be a monophosphate. Okay, so put all that together, we get the prefix deoxy, cytosine. Because it is a pyrimidine, we change the ending to idine, so it becomes deoxycytidine 5' monophosphate. I'm running out of room here. Phosphate. Okay. So rewrite what I had there off. Okay. And of course, if you leave off the 5', a lot of textbooks do that, so deoxycytidine monophosphate is fine as well. Now notice that this name is getting pretty long. So biochemists will abbreviate these names. So for the abbreviation, I'm going to write it here. We're going to have a lowercase d to specify the deoxyribose sugar. So lowercase d in the front, and then a capital, let me underline these, capital C M P to tell you the identity of the base in the nucleotide and to tell you that there's just 1 phosphate. So C tells you that the base is cytosine, And MP tells you that there's just mono, just one phosphate. So lowercase d, capital C, MP, DCMP for deoxycytidine 5' monophosphate. All right, let's do one more naming just for a little extra practice. Let's do probably the best known nucleotide. Let's draw ATP, adenosine triphosphate. Okay, so for one more example, let's do adenosine triphosphate, abbreviated ATP, capital, all capital, ATP. Okay, so what does this tell you? This name tells you that the sugar, let's do it in green again, just to make it stand out. That the sugar must be ribose. Notice, there's no deoxy prefix. So the sugar here is ribose. Okay. The base is adenine. And the phosphates, there's going to be 3 of them, not just one, because this is a triphosphate. There's 3 phosphates here. Okay, so I'll go ahead and draw it in black. I think the black maybe is a little more clear on film. Okay, so let's start with our sugar component, and I'll draw it over here in this corner. Okay, it's a ribose, so make sure you put your OH at the 2' position here. Okay, and then let's go ahead and draw our adenine base. Okay, and if you need to look it up, I do, that's fine. Again, if your instructor wants you to memorize those structures, I feel sorry for you, but then you'll have to do that. But otherwise, just look back to slide 3, and we're going to put our N-glycosidic bond. Okay, make sure I didn't make any mistakes there. Okay, so that is the structure for adenine. Now that I've linked it to ribose, we have adenosine. Now let's add our 3 phosphates. So we've got 1, 2, 3. Okay, so 1, 2, 3. Whoops, made a mistake. Put the oxygen there. Glad I caught that. Okay, 1, 2, 3 phosphates. Okay. So just last but not least, just kind of go over it one more time. We've got the sugar ribose. We know it's ribose because we have the OH at the 2' position, the 2' position. We have the base, adenine, connected to ribose by that beta N-glycosidic bond. And then we have 1, 2, 3 phosphates for adenosine triphosphate, a nucleotide. All right, let's go ahead and erase here and go on to nucleic acids, how these nucleotides link together to make up polynucleotides or nucleic acids. So we've already said that nucleic acids are polymers made up of the smaller units known as nucleotides. So now that we know what nucleotides look like, we are finally ready to talk about how they link together to make up polynucleotides, also known as nucleic acids. So the alcohol functional group off of the 3' position of one nucleotide will react with the phosphate off of the 5' position of another nucleotide, creating a phosphate diester linkage. Let's write that down. Okay. Oops. The OH off of 3' position of one nucleotide will react with the phosphate off of the 5' position of another nucleotide, creating a phosphate diester linkage. Okay, so let's show a picture of how this happens. Okay, so let's pick deoxyribose for our sugar, and let's just keep the base generic in our drawing, and then I'll just write an A here so that we know that that's representing the base adenine, but it takes kind of a long time to draw them all. We're going to be drawing a lot of them here for our polynucleotide chain, so I'm just going to be a little bit generic there. And then we've got our CH2 to our phosphate ester of our nucleotide. Okay, so here's one nucleotide, and let's label, make them stand out, let's label in pink our 1', our 2', our 3', our 4', and our 5' position. So we'll show the 3' OH of this nucleotide reacting with the phosphate off of the 5' position of another nucleotide. So let's draw our second nucleotide here. And let's just make our nitrogen-containing base cytosine. So I'll write a C there, just to be generic. And we'll make it deoxyribose. Okay, and let's number the 1' position, the 2', the 3', the 4', and the 5', and then we're going to show the phosphate off of the 5' position of this nucleotide reacting. So we're going to get a reaction here. In fact, I'll go ahead and just indicate not a mechanism, just a bond formation there, just to kind of give you an idea of what's going to happen here. Okay. And the result will be a dinucleotide so you can see it's a lot to draw, so hopefully you don't blame me for being lazy with my bases, because there's still a lot to draw here, even with that shortcut. Okay, so this phosphate didn't react. That's left intact. But this alcohol off of 3' reacted with phosphate off of the 5' position of the other nucleotide, and we're going to get a phosphate diester linkage. Okay. And again, we made this one a cytosine. And okay, and so let me label. Got our 1', our 2', our 3'. Our 3' alcohol is now gone. It was involved in the reaction. It's now part of the phosphate diester linkage that's connecting these two nucleotides, creating the dinucleotide in this case. 4' and 5'. And then we've got our 1' here, our 2' here, our 3' here, our 4' here, our 5' here, and the phosphate off of the 5' here is now part of that phosphate diester linkage as well. Let's go to the blue pen and let's label that. This is our phosphate diester linkage that connects the two nucleotides and makes this dinucleotide. All right, so you can probably imagine that the free OH at the 3' position here can now react with another nucleotide, the phosphate off of the 5' position of another nucleotide, and then we'd get a trinucleotide, and so on. We'd end up with these very long chains of nucleotides. We'd end up with a polynucleotide, or another word for that is a nucleic acid. All right, so let's go ahead and draw one more just to make it a little bit bigger. Let's show this alcohol reacting with another, has reacted with another nucleotide and let's make the nitrogen-containing base be a guanine. So here's a G. Just put a G there. All right. Now, very important. In fact, I think I'll go to the green pen for this. The free end of the polynucleotide chain with the phosphate is referred to as the 5' end. Okay, so this is our 5' end. And notice it has a free phosphate. The end with the free OH at the 3' position. In fact, let me label these real quick. 3', 4', and 5'. Okay. This is known as our 3' end. The 3' end always has the free OH group. And of course, you can imagine that reacting with the phosphate of another nucleotide, and you can imagine our growing polynucleotide chain. All right, so for this specific trinucleotide, we would label it as 5' ACG for the order of the nucleotides. A, C, G, and then we would end with a 3', so that we would know that it is the deoxyadenosine nucleotide that has the free phosphate in our chain, and that it is the deoxyguanosine side that has the free OH at the 3' position. Okay. So remember when we talked about proteins, how we said that the order of the amino acids is very important. And really, if one amino acid is out of order, the entire protein may not be able to function. The same thing goes for our nucleic acids. In other words, 5' ACG, 3', is not the same, let me write it down here, ACG 3' is not the same as having the sequence 5' GCA 3'. Because in the first polynucleotide chain, we can see that the free phosphate is off of the adenine component, the adenosine, I should say, the deoxyadenosine nucleotide. We can see in the second polynucleotide that the 3-phosphate group is not off of the deoxyadenosine nucleotide, but rather it's off of the deoxyguanosine nucleotide. So they're similar, they're made up of the same 3 nucleotides, but they are not the same. And the order is important for the function of the nucleic acid. Well, it's really important for the nucleic acid to be able to make the proper functioning protein. And that's something we'll be talking about in our future tutorials. All right, another thing I want to point out that is a similarity to proteins is that just as the proteins had a very repetitive backbone, you might remember that a protein's backbone is always carbonyl, carbon, nitrogen, carbonyl, carbon, nitrogen, carbonyl, carbon, nitrogen. And then what hangs off of those carbon units varies based on the side group of the individual amino acids. Same thing goes for nucleic acids. They have a very repetitious backbone. Their backbone is always phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then what hangs off of those sugars can vary because we know we have different bases that can hang off of those sugar components. Okay, so I'm going to indicate that here. So, repetitive backbone sequence of a nucleic acid. Always phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And for a better idea of what that's going to look like, you can look up here. Phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay, but I'm just going to write P's and S's to make this go by a little bit faster. And we know that the phosphate end is always going to be, the free phosphate is always the 5' end, and the free sugar is always going to have that OH, that free OH, that's our 3' end. And then what varies would be what hangs off of those sugars, because that can be different bases. Okay, so I'm going to put little bonds to all my sugars here. And maybe here it's a base adenine. Maybe here it's a base thymine. Maybe here it's the base cytosine. Maybe here it's a guanine. Maybe another adenine and another adenine. But that's what varies, would be your individual bases. The backbone is repetitive. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay. All right, we are definitely out of room here. So let's go ahead and erase, and we will go on to our final slide, slide 6, and talk about the DNA double helix. So we said earlier that DNA contains the entire genetic code for an organism, and it's contained in the nucleus of most cells. DNA is composed of 2 polynucleotide strands running anti-parallel to one another. And those 2 polynucleotide strands are held together by hydrogen bonding. And that hydrogen bonding always occurs between A and T and C and G. No other combination will work. So you're not going to see T hydrogen bonding to C and G hydrogen bonding to A, okay? It always has to be A to T and C to G. Again, no other combination will work. So let's write those points up here on the board. So the DNA is composed of 2 polynucleotide strands running anti-parallel to one another. And those 2 strands are held together by hydrogen bonding. All right, H bonding, which is always between A and T, and I'm going to indicate 2 hydrogen bonds forming between those 2 bases, okay? Because as you'll see in the details drawn out for you on slide 6, adenine and thymine will form 2 hydrogen bonds, and the other pair is C and G. And notice that I made 3 hydrogen bonds here. And again, if you want to see specifically how those hydrogen bonds form, you can look on slide 6 where I have actually drawn those molecules out and I've shown the hydrogen bonding that occurs between those bases. And we call this tendency of adenine to always hydrogen bond to thymine and cytosine to always hydrogen bond with guanine, we call this complementary base pairing. I'm going to write this here. Complementary, oops. And it is fundamental to your understanding of the function of DNA. It is going to be fundamental to you understanding how DNA replication occurs, as well as transcription and translation. So keep this in mind. You need to remember A and T hydrogen bond and C and G will hydrogen bond. That is complementary base pairing. All right. In addition to these details, let's write a few more details up here on the board. So we've got 2 polynucleotide strands. They're running anti-parallel. They're hydrogen bonding to one another. And we're talking about a lot of hydrogen bonding here because there are about 3 billion base pairs in the entire human genome. And then if each base pair has 2 to 3 hydrogen bonds, we're talking about a lot of hydrogen bonding here. So we're talking about a very stabilizing impact on the DNA double helix. Okay, so we've got those 2 polynucleotide strands running anti-parallel, hydrogen bonding through complementary base pairing A to T, C to G. And the 2 polynucleotide strands will actually form a spiraled structure, much like a spiraled staircase, a twisted spiral structure. So I'm going to write here, spirals like a spiral staircase. To make up the structure, the overall structure, I'll say, that we know as the DNA double helix. Okay, so let's draw a picture to try to represent all of this information here. All right, and there's a nice picture in your notes but I'm going to keep my drawings a little bit more cartoonish, a little bit more generic. I'm not going to show the details of the structures of the bases. I'm just going to write them like I've written up here. Okay, so we already know that our backbone will be phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on. And then at our free sugar will be our 3' end, and it will have a free OH group. So I'm going to label this 3', and that our free phosphate is going to be the 5' position. And then we know that our bases will be off of our sugars. Okay, so I'm going to just write the letters. So I'll just pick A, C, and T. Okay. So if the other polynucleotide strand is running anti-parallel to this, then we would have over here phosphate, sugar, so the 5' end down here. Let's see. Let's make that a little bit neater. I need that. Yeah, need the sugar opposite here. Sugar, phosphate, sugar, phosphate, sugar, and then we'll make this our free OH here. Okay, so this would be our 3', and this would be our 5' end. Remember, anti-parallel. And then our bases would be coming off of our sugars, and they have to complementary base pair. So what base must be off of the sugar? Well, adenine only complementary base pairs to thymine, so there must be a T here. And then I'll put my hydrogen bonding in blue, try to make it stand out. What base has to be here? Well, C only hydrogen bonds to G. And then they form 3 hydrogen bonds. Okay. And what base must be here? Well, it's got to be A. Because our complementary base pairing with thymine has to be to adenine. So then I'll put 1, 2 hydrogen bonds there as well. All right. So you can kind of see how that double polynucleotide, those 2 polynucleotide strands, hydrogen bonding, the bases hydrogen bonding between them, you can kind of see how that starts to look like a ladder. And so you'll often see the DNA double helix depicted as a ladder, where the phosphate sugar backbones are the sides of the ladder, and then the bases that are complementary base pairing are the steps of the ladder. And again, these are anti-parallel, so this would be 5' to 3'. That means this would have to be 3' to 5'. And then to understand that spiraled structure, imagine grabbing the 2 ends of that ladder and twisting in opposite directions so that you get the spiraled staircase, which you'll often see drawn kind of like this. Okay, so if this is our 5' end, follow the polynucleotide chain to the 3'. And if this is running anti-parallel, then this must be 3'. Follow it to the end. There is your 5' end. Okay, so that's how I will usually be drawing the DNA double helix in future tutorials, just to save time, using that kind of cartoonish ladder depiction. Okay, spiraled ladder or spiraled staircase depiction. All right, so what I want to do to just finish up the tutorial for today is just do a quick contrast between DNA and RNA. So let's write a couple more things up here for DNA. So we know now it's composed of two polynucleotide strands running antiparallel to one another, held together by complementary base pairing, A to T, C to G, and that it overall has this spiral staircase structure. But a couple more things that we can add here, just review from earlier in the tutorial. Remember that DNA, so I'll add a couple more bullet points down here. Sorry, it's getting a little bit messy. DNA contains the entire genetic code. Remember that it is found within the cell nucleus. And remember that DNA will always contain the sugar deoxyribose. And it will contain only the bases A, T, C, and G. No U. Contains the bases A, T, C, and G. Never uracil, never U. Okay. All right, so that's a summary for DNA. So I'm just going to verbalize kind of the contrasting summary for RNA. So they're both composed of nucleotides linked together in the same way. They're both nucleic acids. But how is RNA different? Well, remember that RNA does not contain the entire genetic code. RNA contains just a copy for one gene. Okay, one gene, a copy of one gene, not the entire genetic code. Remember that RNA is found mostly outside of the cell nucleus, in the cell cytoplasm, because it is intricately involved in protein synthesis, okay, which doesn't occur in the nucleus. It occurs outside of the nucleus. Remember that RNA contains the sugar ribose, not deoxyribose. And RNA contains the bases A, U, C, and G. Because when that copy of RNA is made, everywhere that there was a thymine in the DNA, it's replaced with a uracil in the RNA copy. All right. DNA has two polynucleotide strands, but RNA is going to be single-stranded. It's going to be a little copy of a gene from one polynucleotide strand. So always going to be single-stranded. Okay, so that will conclude our tutorial number 25. I hope that you will consider tuning into problem set 25, where we will talk through lots and lots of problems on nucleic acids. Thank you very much.